After logging into Illumina Connected Multiomics, click on the New Study button. You need to give a name for the study and specify the description. Select an ICA project that stores the files. Click Create Study. After the study is created, click Add Data and choose the files to ingest them to the study. You will get a notification when the ingestion results have succeeded. Now let's create a new analysis. Give a name to the analysis, choose the custom protein option, including all the samples in the study. Click Run Analysis. Once the status of the analysis is changed to complete, you can click on the analysis to go into the pipeline task graph. Mouse over the data node circle. It displays the number of samples and the number of features contained in the data node. Click on the Metadata tab to view and edit sample metadata. Click on Manage under Sample Attributes. You may want to remove some attributes that are not informative. You can change the order of the attributes by dragging and dropping to the left or right. It is a good practice to put the most important attribute at the beginning, since the default color of some plots will use the first attribute to color the sample. Next, we will filter to include only the plasma samples in T1 and T2 time points. Click on the data node, choose Filter Samples. Select to include samples with time point labeled as T1 and T2. Click Finish. The filtered task output has 110 samples. There are control probes in this assay. Next, we will filter to only include the human proteins. Choose Filter Features on the menu. Select Saved List option. Choose a saved list. Click Finish. The normalized protein expression might still have 0 counts, which can cause problems for the downstream analysis. We will perform normalization task. Choose Add 1 to all the values. Click Finish. Next, let's take a look at the distribution of samples. Select Normalize Data Node. Go to Exploratory Analysis. Select PCA. We will use the default setting and click Finish. PCA will reduce the high-dimensional data to 2D or 3D for us to visualize. Double-click on PCA node to open the report. The report displays 3 plots in the data viewer. The scree plot shows the variant represented by each PC. The component loadings table displays the correlation of every feature versus every PC. We're going to focus on the scatter plot. Here, each dot is a sample. The color represents phenotype. Press the left mouse and drag. You can rotate the 3D plot to examine the data from different angles. Click on the Reset button to go back to the default. Scroll mouse wheel in one direction to zoom in. When you zoom in, you can press down the right mouse button to move the plot. Check different areas. Scroll the mouse wheel in the other direction to zoom out. Click on this button to reset the view to the default. The PCA scatter plot allows you to visually identify clustering patterns and outliers. In this data, we can see the samples are clustered based on phenotype on the second PC, which means you will expect some proteins to show differential expression between the phenotypes. The left side is the control panel. Click on Style to customize the plot style. You can choose to use different attribute to color the points, or you can use the size of the dots to represent the different time points. Drag the scale or manually type in a number to change the size of the dots. Click on Grouping. Use the line to connect the samples that come from the same donor. On the top of the control panel, click the Save or Save As button to save all the plots into one data viewer session. To export an image file of one plot, click on the Export Image button. Choose a file format. Click Save. Next, we will detect differential expression on the Normalize Counts node. Click Differential Analysis. Limit Trend is the default method. Click Next. Build a model based on your experimental design. In this example data, we will choose phenotype and time as factors. We will also add an interaction term of the 2 factors. Since it is a paired experiment, we should include the donor ID in the model to avoid violation of the sample independence. Click Next to set up comparisons. We will compare tumor versus normal, regardless of the time points. We also can choose the interaction term to compare tumor versus normal at each time point respectively. Set up all the comparisons you need, then click Finish. Double-click to open the Differential analysis report. The report contains a table of proteins with statistical results. Each comparison generates a section of columns representing p-value, FDR, which is multiple test-adjusted p-value, ratio and fold change in linear scale, and the mean of the two compared groups separately. You can click on the column header to sort the table. By default, the proteins are sorted based on the first comparison p-value. Click on the icon next to the comparison name to open the volcano plot. By default, proteins significantly different in the comparison are highlighted. Click any blank space to deselect. Significant criteria are fold change up or down twofold, represented on the x-axis, and p-value less than 0.05 represented on the y-axis. We can change the size of the dots to make it more visible in the style dialog. Also, you can change the color of the up and down regulated proteins. Click a point to manually select a protein, or draw a bounding box to select multiple dots. Once the points are selected, they will also be highlighted in the bottom table. The top 4 proteins from the table are displayed as a dot plot respectively on the right. In each dot plot, the x-axis is phenotype, and the y-axis is the normalized expression value of the protein. Each dot is a sample. We can configure the plot, for example, to make the x-axis represent time, or make the x-axis represent multiple attributes. Select Categorical List, add both phenotype and a time. We can easily change the order of the attributes using drag and drop. Click on Style. We can change the size of the point, turn off the box and whiskers, or turn off the points and only show the box and whiskers. You can do the same thing for the other genes. On the left side of the analysis report, you can specify certain criteria to filter proteins. We will use FDR and fold change criteria to generate 3 protein lists based on the 3 comparison results individually. Select FDR, choose per contrast, and specify 0.05 only for one comparison. In fold change section, also choose per contrast. Specify -2 and +2 for only one comparison. Press Enter. The number of proteins that passed criteria will be displayed on the top. When you click Generate a filtered data node, the task will be performed on the analysis task graph. Repeat the same steps to generate the other 2 lists of proteins showing difference between tumor versus normal at each time point. After all filter tasks are done, there are 3 filter feature list nodes added to the pipeline. You can right-click on any data node to rename it. To compare the 3 lists, click on Venn diagram and select the lists. Make sure they are using the same identifier. You can select up to 5 lists to draw the Venn diagram. Click Display Selection. Here's the common proteins that are in all 3 lists. Click on any section. The proteins in that section will be displayed on the right side. You can download it or save it, or use it to filter your data node on the pipeline. Let's use the tumor versus normal feature list as an example to visualize the results. We will use a heat map to show those proteins that are different between the 2 groups. Click on the data node, go to Exploratory Analysis, and choose Hierarchical Clustering Heat Map. Select how you want to order the proteins. When choosing clustering, the order of your proteins will depend on the clustering result. For the samples, let's manually assign the order based on phenotype. We can rearrange the sample group using drag and drop. Click a Finish. Double-click to open the heat map. By default, samples are on rows, proteins are on columns. Let's go through the configuration here. Click on Axis. We can turn off the sample name labels. Make the column labels only show gene symbol. In Heat Map dialog, You can change the color. Default blue represents low expression values, red represents high expression values. Click on the little color tab to change the color. You can add a color in between. Suppose we use red to represent high expression and pink to represent extremely high outliers. Slide the scale to change the contrast of the colors. In the dendrogram dialog box, you can change the height of the dendrogram. Annotation allows you to add sample attributes. Click the Transpose button on the right. You can transpose the plot to put samples on columns and proteins on rows. Zoom in and out by scrolling the mouse wheel. Let's find out if the filtered protein list is overly represented in any pathways. We will go to Biological Interpretation, click Gene Set Enrichment Task. Specify the database and click Finish. Open the pathway enrichment for our report. Each row is a pathway. Columns are the analysis results. Enrichment score is converted from p-value. You can download genes in each pathway. Click on a pathway name to view the gene network. If you want to detect which pathway is showing differential expression between tumor versus normal, you will click on the Normalized Counts data node. Choose Biological Interpretation. GSEA is a popular method to answer this question. Choose Database and click Next. Select Phenotype to compare tumor versus normal. Click Finish. In the report, each row is a pathway. They are sorted by normalized enrichment score. Click on the enrichment plot to visualize this particular pathway. To conclude, in this video, we learned how to create a study to analyze Illumina Protein Prep data in Illumina Connected Multiomics.